Thank you for standing by and welcome to the Chimeric Therapeutics Core NK Phase 1 Data Investor Webinar. Apologies for the short notice as we were getting the announcement onto the ASX platform this morning, but this session will be recorded for viewing at a later point for anyone else. All participants are in a listen-only mode. There will be an initial presentation lasting for approximately 15 minutes, followed by some Q&A. If you have a question you'd like to submit, please feel free to type it in using the Q&A function on the Zoom app. Presenting on behalf of Chimeric Therapeutics today, we have CEO and Managing Director, Jennifer Chow. I'll now hand it over to Jen. Please go ahead.
Thanks so much, Matt. And thank you everybody for taking the time, even with some short notice there, to join us this morning to hear the results. A little bit of irony, as I am noting that my very first meeting in Australia today is actually by Zoom, but we'll go ahead anyway, and I look forward to meeting many of you later this week. So just a quick reminder of where we are, or what the Core NK platform is, so the data that we're talking about. The Core NK platform is a universal off-the-shelf NK cell platform technology. And that essentially means that we take cells from a healthy donor and we enhance them and then we freeze them, and we're able to then provide them to patients all over the world. This particular platform was developed and invented by Dr. David Wald at Case Western, who's actually won many awards for his work on novel feeder cell lines. And really, as he developed the Core NK platform, he did so using all his expertise and his knowledge to overcome two very specific concerns about NK cell development. The first is what we call expansion. And so that's really about being able to manufacture and generate enough really enhanced active cells to give to patients so that there is enough cells to overcome the cancer. And then the second is persistence. And that's really about making sure that when the cells get into the body, they stay alive. So those were really what Dr. Wald was focused on enhancing in this platform as he went forward. And you'll see in our trial that he actually has been able to achieve those objectives, as well as a number of other exciting endpoints. Chimeric took the exclusive option to this platform in December of last year, and we're now moving ahead with full licensing and a research agreement with Dr. Wald to continue the development. So without further delay, and although lots of you have probably seen this with the ASX announcement, this is the highlight of the data set that has just been published. And so we're incredibly excited because this data set we actually believe would probably be published later this year, and we've had an accelerated publication of it. What we saw was very, very compelling efficacy in blood cancers, specifically high-risk MDS and AML. There were 3 patients treated that had blood cancers. All 3 of those patients achieved disease stability for 100% disease control rate. Now, 2 out of those 3 patients actually maintained their response more than 100 days. And very, very notably, 1 patient actually deepened their response to have a complete response. So just 2 doses of the NK cells, and this patient had a complete response that's now an ongoing response past 15 months. So really exciting and compelling data that we saw with the blood cancer patients. On the solid tumor side, we also saw what we believe is very promising data. There were 6 solid tumor patients treated, and we actually saw 2 of those 6 patients have disease control as well. So really, from an anti-tumor effect or efficacy effect, we're really excited and pleased by what we're seeing, particularly in the blood cancers. And with the solid tumors, this is data that we anticipated. And I'll talk to you a little bit about how we really are going to look to move this forward with this data. The primary objective for the trial was safety. And so we were really excited about being able to see that there was no graft versus host disease. And this is really important with allogeneic or off-the-shelf platforms, because we're using cells that are donated by one person and we're giving those cells to another person. And so what we really want to be sure is that the person who's receiving the cells, their body doesn't reject the cells. So that no graft versus host disease means that we didn't see any of that. And that was really important to establish with these cells. We also saw no cytokine release syndrome and no dose limiting toxicities as well. And then Dr. Wald was able to meet those goals of establishing both expansion and persistence of the cells. So robust expansion, we had a 21-day manufacturing that gave us over thousands of doses of cells available for patients. And we were able to see persistence of the cells more than 4 weeks. So very, very positive data coming out of this phase 1A trial. Just to give you a little bit of background on the trial itself. The trial was open for a broad group of patients, both blood cancers and solid tumors, and you see the list there. And the primary objective obviously being safety, the secondary objectives being to look at the GVHD and the anti-tumor effect. And then the study actually started in 2018. The study was completed in mid of last year in 21. And as I mentioned, we're really pleased to be able to share the results now, as we had anticipated it wouldn't be a few more months before we saw this. From a trial design, it's a simple, what we call 3x3. So essentially there were 3 different dose levels in the trial. 10 million, 25 million, and 50 million cells per kilogram were the 3 different dose levels. And in each dose level, 3 patients were enrolled. Across the entire trial, there were 3 patients with blood cancers and 6 patients with solid tumors. And you see in the simple schema below how patients actually received the NK cell therapy. They had a lymphodepletion, and then on day 0, they received 1 infusion of the NK cells, or the core NK cells. And then on day 14, they received a second core NK infusion. At day 28, we actually looked to see whether or not the patients had had any response. So we did a disease assessment. And then at day 100, we did a second disease assessment to see whether or not any of the patients that had a response had been able to maintain the response. So very simple phase 1A trial design with this trial. As I mentioned, the safety that came out of it was very, very exciting to us. No graft versus host disease, no cytokine release syndrome, no dose-limiting toxicities. And this really allows us to think about whether or not we start to look at this platform at higher doses as well, because we certainly really were able to establish good safety at these doses. From an efficacy perspective, now this gives you all of the details here. I'm not going to go through all of them. I'm going to highlight them a little bit, but generally what we saw across all 3 dose levels and the 9 patients was good disease stability. About 56% of overall patients achieved disease stability, including very notably this 1 complete remission we saw, and I'll talk about. In blood cancer patients specifically, this is just the 3 patients that had blood cancers. And you see these patients were generally treated with the lower cell doses, the 10 million and 25 million cells. They all had MDS, AML, high risk circulating blasts, and they'd all had multiple previous lines of therapy. All 3 patients that had blood cancers achieved at day 28 a response of stable disease. So really, really exciting. What we saw then was 2 patients go on even further. One of the patients had that stable disease last all the way through to the day 100 assessment. And then the second patient actually had what we call a deepening of the response. And by day 100, actually had gone into a complete response. So incredibly exciting for us to see this at these dose levels in a phase 1 trial. That particular patient that had that complete response at 15, that complete response actually now has been in remission for over 15 months. This just gives you a little bit more detail about that patient. They had high risk MDS with circulating blasts and a high tumor burden. They have progressive disease coming into the trial. They'd had a previous allogeneic transplant. Stable disease, as I mentioned, by day 28, and by day 60, they'd actually deepened to a complete response. From a sustained complete response at day 100, the patient actually received a consolidation transplant, and now 15 months later continues to be in remission. So really, really exciting what we're seeing here with this patient. In solid tumors, as I mentioned, we saw disease control in 2 of the 6 treated patients. Now here, all of the patients generally had either colon or colorectal cancer. They all had multiple previous lines of therapy, and they all had pretty extensive metastatic disease. With this, one of the 2 patients that had disease control at day 28 actually went on to continue to have the disease control or disease stability at day 100. And so this is really what we anticipated seeing in blood cancers versus solid tumors, is that there would be a better response in patients in blood cancers than in solid tumors. What we do know in solid tumors is that we have a number of planned development paths to really be able to enhance these responses. We can add exogenous cytokines. We can look to add tumor bed infiltration mechanisms like dominant negative TGF-beta. And then we also can look at being able to put this with our existing chimeric antigen receptors. So we're seeing a response here. We're seeing a response with our CARs like fluorotoxin. And then we are now looking at how we bring that together and really amplify the effect, particularly for these solid tumor patients. We also were able to establish some really key things about this platform. We used a universal donor, and importantly, we did not do any HLA matching. And so this is a true universal off-the-shelf therapy where we've taken the cells from one healthy donor, and that one donation of cells, we expanded through manufacturing for 21 days. We were able to get up to 30,000-fold cells, and that actually gives us thousands of doses that we can use for patients. And then when we put the cells into the patients, we actually did see that important 28-day persistence with no rejection of the cells. So really important, some of the really important basic science and translational work we were able to establish here. So bringing it all together, so there's a lot of data in this trial and in the publication. I certainly invite you, the references down is here, and invite you to go and take a look at the publication. There was 5 very important things that we were able to establish here that we were able to show we completely were able to overcome the challenges of NK cell development. The first for us, very exciting, was that therapeutic effect in the blood cancers and solid tumors. The second was the safety with the universal donor cells. The third, this 28-day persistence. The fourth, the robust expansion. And the fifth were the lessons that we've now learned about how to amplify the therapeutic effect of these cells. So now we're really focused on accelerating all of the development paths that we'd originally identified for this platform. We're going to look right away at rapid development as a combination therapy in blood cancers with the existing platform. And then in parallel, we're building out a next generation platform to add some additional enhancements to. And then for those solid tumor patients, really looking at adding in our gene editing in or genetically modifying the NK cells with our CARs so that we're able to better address the needs of solid tumor patients. And then we were already having discussions about how to leverage this platform for collaborations and out-licensing, and certainly know that this data is just going to really open up our ability to do that even more. So very exciting for the future. So with all of that, we're coming back together here in terms of what the overall portfolio and pipeline looks like. We have those 7 assets now in development with 8 clinical programs across at least 10 different disease areas. And certainly when you look at, the portfolio, the novel assets in there across multiple disease areas, the extensive clinical development is really exciting, and thrilled to now be able to go from saying early positive signals in one trial to being able to say early positive signals in multiple trials in multiple different types of cancer. We have the world-renowned partners and collaborators that we continue to work with to drive these forward, and certainly our team that is really focused on being able to bring these assets through development to get them to patients as soon as possible. So with Matt, I'll stop and see if there is any questions.
Thanks very much, Jen. Just to repeat for everyone, if you do have a question, please type it in using the Q&A function within the Zoom app, and we'll get through as many as we can. Jen, the first question is, did the dose levels in this trial seem low compared to the chlorotoxin trial? Can you provide some comment around this?
Yeah, so dose levels in any trial really are very specific to the asset. So this is a very different therapy than the chlorotoxin trials. Chlorotoxin is a T cell derived therapy that is autologous. This is an NK cell derived therapy that is off the shelf or allogeneic. And really what we do is we look at the preclinical work and then really set where the dose levels are. So, I think the other thing to keep in mind is that chlorotoxin is flat dosed and this is dosed by weight. So when you're looking at 10 million cells, it's 10 million cells per kilogram. So in actual fact, there is definitely differences in dosing, but really have to do with the assets and what we believe is needed to really affect patients.
Thanks, Jen.
Yeah.
The next question is, what would have been the disease progression prognosis for these patients in the absence of this therapy?
Yeah, that's a great question, Matt. Little difficult to answer because it was all different disease types that were coming in. So an AML patient or an MDS patient that, if I use the patient that had the 15-month remission as an example, really that patient had had 3 or 4 previous lines. They'd had a previous transplant already. The patient sadly would be running out of options quite quickly. And with a disease that aggressive with circulating blast and a high tumor burden, you probably were looking at 3 to 6 months. It probably is a little different, Matt, in some of the other patients, and it really would have to do with what other agents they'd been predisposed to. But probably for most of these patients, they had tried multiple previous lines of therapy. They really were coming down to sort of some of the last attempts, and probably that 4 to 6 months is probably what we would have expected to see for a lot of them.
Great. Another question that's come through is, considering the large number of excellent opportunities Chimeric has to develop, how are you going to prioritise these moving forward?
Yeah, another great question. Thanks, Matt. I think from the NK cell therapies, what we're looking at in terms of prioritization is there is sort of a number of things we can do in parallel, and we will be looking to do in parallel. We obviously now have an NK cell platform that's been proven, and so we can look to move this into different trials right away, while at the same time, we do some of the lab work to further enhance the platform and obviously build our CAR NKs. So there is definitely some work that we can easily do in parallel, Matt, and then there's definitely some things that will take, we have to do sequentially. So from a priority perspective, really from our side, with this particular program, we really want to be able to move forward with now seeing this data with next steps with this platform, and in parallel rapidly move the development in the lab that Dr. Wald will be doing. So luckily that will take us, different groups doing different things, and we'll be able to move them both forward quickly.
Thanks, Jen. And a somewhat related question, do the results from this phase 1 trial you've announced today change the, the goalposts moving forward in terms of how much attention you're going to give to this program?
I don't think so, Matt, because to be quite honest with you, we were always very, very excited about this program, right? So not to say that this isn't amazing data, and particularly seeing this CR for all of us has really been really astounding. But I think that at least for our small team, we believed in this platform and Dr. Wald's work, or we wouldn't have brought it into the company. We knew it had this type of potential. So I think it doesn't change our drive or our efforts to develop it further. Perhaps it does accelerate some of our work, because I think there'll be a lot of attention paid to it now from external forces and collaborators. But certainly this for us is really just reinforcing the belief we already had in the platform.
Great. Well, with that, I don't have any more questions in the queue. Jen, I don't know if you just wanted to provide a final comment before we wrap up.
Yeah, very much so. Thank you, everybody, for taking the time. Certainly, if there's any other comments or questions, happy to take them offline, and the contact information is always available. We are incredibly excited about the results that we have seen here. We believe it does, like I said, reinforce what we believe the potential for this platform is, and really believe that, as we said when we brought it into the company, it's going to be transformational for what we're doing. So really exciting days ahead. Thanks, Matt.
Thanks, Jen. And I probably should also add to that, as Jen alluded to at the beginning, Jen's in Australia this week. There's a presentation, speaking of short notice, at 4:30 p.m. for shareholders in Sydney today. And then there's one in Melbourne at 4:30 p.m. tomorrow. If you would like to join those, please feel free to reach out to myself or Jen on the contact details on our announcements, and we'd be happy to have you along. But with that, we'll leave it there for today. And thanks to everyone for joining, and thanks, Jen, for the presentation.
Thank you. Thanks, everybody.